This is Professor LVKS Bosker. In this video, I am going to explain various concepts involved in the origin of life. Chemogeny, which is also referred to as chemical evolution. This concept involves the synthesis of complex organic compounds, an essential step in the development of life. The process unfolds in several steps. Synthesis of simple organic molecules. These serve as the building blocks for more complex compounds and mark the origin of organic chemistry. Formation of complex organic molecules. These are larger and more intricate molecules formed from the simpler ones. Formation of nucleoproteins. These proteins are crucial as they combine with nucleic acids and contribute to life structure and function. These organic compounds are vital for both the structure and function of living organisms. The process described involves the synthesis of simple organic molecules from the basic elements carbon, C, hydrogen, H, nitrogen, N, and oxygen, O. As Earth's surface cooled, these elements combine to form compounds like water, HO, ammonia, NH, methane, CH, and cyanide, CN. When the Earth's temperature dropped to around 100 degrees C, reactive free radicals such as CH and CH condensed, leading to the formation of various hydrocarbons. This process of molecular formation was driven by condensation, polymerization, oxidation, and reduction reactions. This explanation highlights the basic principles behind the formation of organic molecules on early Earth. The next step in the formation of life involved reactions between these simple molecules, formed in earlier stages, in hot water, which led to the creation of more complex molecules such as purines, pyrimidines, and nucleotides. These are essential building blocks for the formation of DNA and RNA, key molecules for life. The energy required for these reactions came from various sources, including UV radiation, electric energy from lightning, heat from volcanic activity, and the Earth's own temperature. The result was a mixture of organic compounds in the hot sea water, a concept known as the hot dilute soup or prebiotic soup, coined by JBS Haldane. Importantly, there was no free oxygen, O, present on the early Earth, which contributed to this unique environment. Biological evolution, also referred to as biogeny, is essentially the process through which species change over time. This happens because of changes in the genetic material, DNA, of a population. For evolution to occur, these genetic changes need to be passed from one generation to the next, meaning they are inheritable. These changes can be small, but accumulate over time, leading to noticeable differences. These genetic changes are reflected in the population's phenotypes, the observable traits like size, color, or shape. As evolution progresses, these physical traits start to shift, which shows that genetic evolution is taking place. So, biological evolution drives the diversity and adaptation of species, enabling them to survive and thrive in different environments over generations. Formation of DNA and RNA components, basic building blocks like sugar, phosphate, purines, and pyrimidines combine to form the fundamental units of DNA and RNA. These nucleic acids are essential for storing and transmitting genetic information. Conservates and intermediate life forms. These nucleic acids, along with other macromolecules in the primordial soup, led to the creation of intermediate life forms called conservates. Conservates are simple, cell-like structures that can absorb organic material and grow. Formation of protobionts. The conservates absorbed more organic material, grew larger, and eventually gave rise to the first cells. Russian scientist Alexander Oparin called these early cells protobionts, marking an important step in the origin of life. This sequence illustrates the early stages of life's evolution, where simple molecules combine to form complex, self-replicating systems, which eventually developed into the first cellular life. In early Earth's oceans, simple organic molecules reacted in hot water, forming essential compounds like purines, pyrimidines, and nucleotides. These molecules are the basic building blocks for DNA and RNA, vital for life. Energy for these reactions came from UV radiation, lightning, volcanic heat, and the Earth's temperature. The mixture of organic compounds in hot sea water is referred to as the hot dilute soup or prebiotic soup, a concept proposed by JBS Haldane. This environment allowed complex molecules to develop under conditions devoid of free oxygen, O, which is crucial because oxygen would have disrupted these early processes. The theory describes how Earth's early oceans acted as a chemical nursery, providing the right conditions for molecules to combine and evolve toward the formation of life. This period represents a critical phase in the prebiotic chemical evolution leading to the origin of life.
The RNA world hypothesis proposes that RNA was the original genetic material due to its ability to self-replicate, allowing it to carry genetic information across generations independently. This theory has spurred considerable debate among scientists for the past 50 years. Many experts agree that life could not have emerged directly from non-living chemicals in a single step, suggesting that intermediate precellular life forms likely existed. Within the various models of precellular life considered, the RNA world hypothesis remains the most favored. It posits that early life utilized RNA not only for genetic storage but also for catalyzing biochemical reactions, paving the way for the evolution of more complex life forms, including DNA-based organisms. This hypothesis helps bridge the gap between simple chemical precursors and the complexity of cellular life, highlighting RNA's pivotal role in the origins of life on Earth.
The discovery of ribosomes, RNA molecules capable of catalyzing chemical reactions, provides significant support for the RNA world hypothesis. A compelling argument for this hypothesis is that the ribosome, responsible for assembling proteins, is itself a ribozyme. Although ribosomes contain both RNA and protein components, the actual catalysis of protein synthesis during translation is performed by the RNA within the ribosome, not by proteins. This suggests that early life forms likely utilized RNA to catalyze biochemical reactions, before proteins evolved to take on this role. The existence of ribosomes implies that RNA was not just a passive carrier of genetic information, but also an active participant in essential cellular processes. This reinforces the idea that RNA was a critical component in the early stages of life, supporting the notion that life may have originated in an RNA-based world before the emergence of DNA and protein-based systems. Thanks for watching.